In this experiment, my lab group was tasked with collecting bacteria samples from areas across the MC campus using cotton swabs, sterile water, and an agar plate. Dr. Brigatti told me it's often difficult to disinfect fabric underneath its surface levels, so I decided to take my sample from the blue fabric chair in the lobby of Gibson Hall. Our samples were purified via PCR and electrophoresis to induce amplification of the 16S ribosomal RNA gene sequence, a phylogenetic marker present in all bacterial species. My samples, labeled U35 and UD35 to differentiate between the primers used, did not experience amplification during electrophoresis. Pipetting errors during PCR preparation, a lack of sufficient DNA in the sample, a conflict between the DNA and primer designs, or degradation among any of the necessary PCR components could have caused this. Samples with successful amplification were PCR purified and sent to a specialized facility at the University of Tennessee for gene sequencing. I was tasked with analyzing and editing the resulting electropherograms via Chromos software, which gives a visualization of each sample's 16S ribosomal RNA gene sequence as both a string of nucleotide bases and as a series of peaks showing the frequency of each base at points along the sequence. Ideally, we expect tall, narrow peaks because wide, abnormal peaks are less accurate to the actual frequency of nucleotide base present in the gene sequence. I cut these out along with ambiguous bases in which the program couldn't identify a single nucleotide base, which are adenine, thymine, cytosine, or guanine. Once edited, the sequences were compared to those available in the National Center for Biotechnology Information's database to produce max score and percent identity values. Max score represents the average of matched nucleotides and the mismatches or gaps between a sample's gene sequences and the sequence it is being compared to. Percent identity quantifies the percentage of the sample's nucleotide sequence that matches with a given sequence from the database. Most samples received one presumed identity based on a high max score or percent identity match, but some were given two because they matched with different species between the sequences using forward primers and those using reverse primers. This overlap between species is the main reason I believe further experiments should be conducted towards perfecting a more accurate method of bacterial identification.